Okay, I really want to hammer home this point because we're talking about the origin of life as the origins of information. And what does that mean for there to be an origins of information? Well, so in information theory, there's something called the Shannon entropy, which is essentially the expected value of information in a sequence, in a sequence set of symbols formally. And DNA is actually a set of symbols because it's just the alphabet as size 4. There's A, T, C, and G, right? So the Shannon entropy is P is you sum over the probability of each symbol, where Pi is so the ith symbol has a probability Pi. You just sum over all of them, and that gives you the Shannon entropy, and we call it H. So that's the Shannon entropy, and I want to narrow that down. And this quantity is maximized. This function has a maximum entropy if the distribution of symbols is uniform. So let's say the symbols, the random variable that denotes the sequence is denoted X. If it's a uniform distribution, meaning each symbol is equally probable, then the H is at a maximum. And so what that means is that if you have a uniform distribution of, so every sequence then has a probability of 1/4 to the K. That is maximum randomness. So this is our definition of what abiotic is. We now have a definition. It's that each symbol, each sequence in the 4 to the K space has an equal chance of being observed. And that's randomness. That's pure randomness. So what life is a deviation of that, from that. It's essentially non-random, meaning deviation from uniform. And that was sort of the idea that my mentor Alan Moses and I worked on in our paper where we proposed the RNA condensate model. And so life is essentially, we're defining it as a deviation from a uniform distribution. And you could visualize this.
Yeah, how does it look like?
So yeah, if you just draw out a uniform distribution, each symbol, it's essentially just like a box, right? So that's a uniform distribution. This is the probability of X. That's X. So that's a uniform distribution. It has maximum entropy. That means each of the 4 to the K sequences is equally likely, right? Okay, well now, what is life? Well, life is when this distribution shifts to particular parts of that space. So let's draw it like this. So this is not exact, but I'm just giving you a rough idea that like when certain sequences are favored, you have a deviation, and then this gives you actually an exact number of bits of information added by a non-random, non-abiotic distribution of sequences. And this information has with it a cost, because no information can be created without energy. And energy and information are proportional, actually. And in our framework here,
and this comes from like the conversion of bits to entropy, correct. Basically.
Bits to entropy. So for every number of bits, there's a certain number of joules. Actually, for every bit has with it Boltzmann's constant times temperature times the log of 2 number of bits, I think. Sorry, number of joules. That's right.
Per binary bit.
Yeah. And that's related to Boltzmann's the KB times ln of the number of states, and a bit you could think of as a system with 2 states. So that's where you get this fundamental relationship, and it's the fundamentals of like we're getting to like what is life. Life is information, and we're defining what it is right here. So okay, so that's that. Now, how do you get that? Well, we've actually formalized the problem now. We've defined what you need, right? Which is you need to have a gain of information over a random background. And yes, and so to get that, one thing you would need is to have replication of a master sequence. So this is from Eigen, Manfred Eigen's framework, is that you have this like parent sequence from which everything descends from, and there's a certain error with every round of replication. Let's call the error rate R, and R cannot exceed 1/k, where K is the length of the sequence. And turns out, so that if you use that, you now have a distribution of errors. And in computer science, when you have strings of equal length, the number of positions in which they differ is called the Hamming distance. And so it turns out the Hamming distance can be modeled as a random variable, and that follows a binomial distribution. So you could think of it as if there's k symbols, each one has a probability r of being incorrectly replicated, and so it's a binomial it follows a binomial because it's yes or no k times. So it's just k choose r times r to the k times 1 minus r to the power of k minus d, and that's the binomial for it. And if you use that and you combine that with another thing is, if you have a sequence of length k, and you have a certain if you have a sequence of length k, the number of possible sequences that are d symbols different from it has a specific form. It's k choose d times 3 to the d. And if you use that and you combine it with the binomial, you can now actually derive what p of x is. And you could actually quantify how different p of x has to be from the background distribution, which is the abiotic distribution. So you have an abiotic distribution, which is uniform, and then you have the non-abiotic, so the biotic distribution, which is non-uniform, and it follows this sort of like, it's not a binomial, but it's related to a binomial. It's a binomial multiplied by the Hamming distance.
One of the things when we were discussing this yesterday, one of the things that I found interesting is that you mentioned is that, and this is just like bringing it back to like empirical stuff, is that the human genome is roughly 3 billion base pairs, and the error rate of copying is roughly 10 to the 9th, so it's 1 in a billion. So actually, that 1 over k number for the error rate threshold, the human genome is like three times lower than that threshold. So it's below the error rate threshold. That's theoretical. It's below. But interestingly, there are plants that are above, like a factor of 100 above the error rate threshold, roughly. But they might contain a lot of junk DNA that, like, when it gets copied, if there's an error, it doesn't actually affect the outcome as much. So that might be the explanation. But interestingly, there are things that actually break the break this rule.
Well, okay. So I'll elaborate on that. So yes. So it's true that plant genomes have error rates that might not fit clearly into this framework, but I'll explain. So sort of like two key things. So first key thing is that in the human genome, the error rate of DNA polymerase is about 1 in a billion, and the human genome is 3 billion bases approximately. And so therefore the error rate is, so again, the Eigen gives you the max error rate. So we're still within the good limit essentially. But when is it okay to pass the error rate? Well, it's okay to pass the error rate if not all the sequence is functional. So this is sort of like a key point. So we could think of the effective number of sequence of bases that are functional. So you could sort of like, think about it as if you're trying to like copy a book. When you copy a book, do you necessarily need the author's letter, or do you need the epilogue, or do you need the acknowledgement section, the cover? Yeah. So like these are the less essential parts of it. And so Eigen's threshold really applies. So there can still be life as long as the functional stuff is okay.